Have you ever wondered what we are all made of? All life is made of cells. They come in thousands of shapes and sizes. The human body has over 100 trillion of them, carrying out the dance of life. They are so tiny that 10,000 could fit on the head of a pin. The cells in our body undergo over 500 quadrillion chemical reactions every second. Their amazing biochemistry has nurtured Earth for billions of years and protected us from harm. They ask only that we take care of ourselves. Our science has barely begun to understand them. Let's explore cells.
Hello students, this is our second lesson and this is all about the cell, the fundamental unit of life. So to start with, what is cell? Now cell is the basic structural and functional unit of living organisms. In other words, cells make up living things and carry out activities that keep a living thing alive. Based on the cell theory, so when we say cell theory, it's a collection of ideas and conclusions from many different scientists over time that describe cells and how cells operate. Number one, all known living things are made up of one or more cells. All living cells arise from pre-existing cells by division. The cell is the basic unit of structure and function in all living organisms. We have here cell theory timeline. 1665, Robert Hooke discovered cell while looking at a thin slice of cork. In 1674, Anton van Leeuwenhoek laid the foundation of plant anatomy and became an expert on animal reproduction. He discovered blood cells and microscopic nematodes and studied the structure of wood and crystals. He also made microscope to view specific objects. In 1883, Robert Brown, he is a Scottish botanist best known for his descriptions of cell nuclei and of the continuous motion of the minute particles in solution, which came to be called the Brownian motion. In 1835, we have Felix Duhardin made his largest contribution to the cell theory when he proposed the classification of a new type of single-celled organism which he called rhizopoda, which are now known as protozoans. In 1838, we have Matthias Claden proposed all plants are made up of cells. And in 1839, we have J.E. Purkinje named fluid content of the cell protoplasm. Now, in 1839, Theodore Swan proposed all animals are made up of cells. In 1845, we have Karl Heinrich Braun revised the cell theory with this interpretation that cells are the basic unit of life. In 1855, we have Rudolf Virchow proposed all cells arise from pre-existing cells. There are organisms that are made up of only one cell, and these organisms are called unicellular. We have here an example, a euglena, a paramecium, and yeast. An organism that is made up of more than one cell is also called multicellular organism. Examples are plants, animals, and fungus. So this is the picture of an multicellular organism under a microscope. Okay, the picture of the leaf cell and a muscle cell. Now, cell size vary in terms of most cells are very small, microscopic, and some may be large or macroscopic. Now, the unit used to measure the size of the cell is called micrometer. So, when we say micrometer, 1 micrometer is about 1 over 1,000 millimeters. Now, the smallest cell, we have the mycoplasma, which is about the size of 0.1 micrometer. So, mycoplasma is a genus of bacteria that lack a cell wall around their cell membranes. And we have also the largest cell, which is in the form of ostrich egg. So, the size is about 18 centimeters. Now, female sex cells are just as complicated. Now, the largest egg produced by a living animal belongs to the ostrich. It is 20 times heavier than a chicken's egg and measures roughly about 15 centimeters tall and 13 centimeters wide. So this is the size of the cell in humans. So the smallest cell, the sperm cell, about 5 micrometers. The largest cell, which is about 120 micrometer in a form of the ovum or the egg cell. And we have the longest cell, which is the nerve cell, that is about 1 meter. So cell also vary in shape. Now, variation depends mainly upon the function of cells. Some cells, like euglena and amoeba, can change their shape, but most cells have a fixed shape. So, human red blood cells, example we have here, are circular biconcave for easy passage through human capillaries. This one is the nerve cell, or the nerve cells are branched to conduct impulses from one point to another. Now the other one, the human WBC or the white blood cells, can change their shape to engulf the microorganisms that enter the body. So the detailed structure of cell has been studied under compound microscope and electron microscope. Now, certain structures can be seen under an electron microscope, and the structure of a cell as seen under an electron microscope is called the ultrastructure. Now, the compound microscope can actually magnify up to 2,000 times, while the electron microscope magnification is about 500,000 times. So, the different parts of the animal cell. So let's have an overview. Here's the nucleus. Another one is we have the Golgi body. We have here the vesicle. This one is the plasma membrane. We have the mitochondria. We have the cytoskeleton. We have the centriole. We have the lysosome. We have the cytoplasm, the rough ER, the smooth ER, and we have the nucleolus. For the plant cell, we have, of course, the nucleus, we have the Golgi body, we have also the vesicle, we have the lysosome, the plasma membrane, the mitochondria, we have the chloroplast, we have the cell wall, the vacuole, the smooth ER and the rough ER, and together with the nucleolus. For the bacterial cell, we have the capsule, okay, we have the cell wall, we have the plasma membrane, we have the cytoplasm, we have the flagellum, we have the food granule, the plasmid, which contains the DNA, we have the ribosome, the nucleoid, and the pili. So if we study a cell under a microscope, we would come across three features in almost every cell. And these three features are the plasma membrane, the nucleus, and the cytoplasm. So all activities inside the cell and interactions of the cell with its environment are possible due to these structures. So let's start with the plasma membrane. So the plasma membrane is extremely delicate, thin, elastic, living, and semi-permeable membrane. So made up of two layers of lipid molecules in which protein molecules are floating. So in terms of thickness, this varies from 75 to 110 angstrom. Okay? This is angstrom. So angstrom is about 1 times 10 to the negative 10 meters. So functions, first, maintains a shape and size of the cell, protects internal content of the cell, regulates entry and exit of substances in and out of the cell, then maintains homeostasis. For the cell wall, this one is a non-living and outermost covering of a cell. So it is also found in the plant and the bacterial cell. Can be tough, rigid, and sometimes flexible. So made up of cellulose, hemicellulose, and pectin. So may be thin or thick, multi-layered structure, and the thickness varies from 50 to 1000 angstrom. In terms of its function, this provides definite shape, strength, and rigidity, prevents drying up of cells, helps in controlling cell expansion, protects cell from external pathogens. The nucleus. So the nucleus is a dense spherical body located near the center of the cell. The diameter varies about 10 to 25 micrometers. Present in all cells except in the red blood cell and the Schiff tube cells. So the red blood cells or the RBC lacks nucleus. It's have a well-developed in plant and animal cells, underdeveloped in bacteria and the blue-green algae, and most of the cells are uninucleated, meaning having only one nucleus. So few types of cells have more than one nucleus. The best example, we have the skeletal muscle cells. So the nucleus has a doubled layer covering called the nuclear membrane. Okay, the nuclear membrane, and the nuclear membrane has pores of diameter, okay, pores of diameter about 80 to 100 nanometers. So colorless dense present inside the nucleus known as the nucleoplasm. The nucleoplasm contains round-shaped nucleus and network of chromatin fiber. So fibers are composed of the DNA or the deoxyribonucleic acid and the protein histone. So these fibers condense to form chromosomes during the cell division. Now the chromosomes in the nucleus, so the nucleus contains the chromosome. Now the chromosome contains the stretches of the DNA, and these are called genes. Now, genes transfer the hereditary information from one generation to the next. So the function of the nucleus is to control all the cell activities like metabolism, protein synthesis, growth, and cell division. So nucleolus synthesizes RNA to constitute ribosomes and also store hereditary information in genes. Next one is the cytoplasm. So the cytoplasm is the jelly-like material formed by 80% of water. Present between the plasma membrane and the nucleus, contains a clear liquid portion called the cytosol and various particles. So the cytosol is the liquid found inside of cells, and it is the water-based solution in which organelles, proteins, and other cell structures float. So the cytosol of any cell is a complex solution whose properties allow the function of life to take place. So particles are proteins, carbohydrates, nucleic acid, lipids, and inorganic ions. So also contains many organelles with distinct structure and function. And some of these organelles are visible only under an electron microscope. Now granular in dense in cells, animal cells, and thin in plant cells. So the endoplasmic reticulum. So we have two types, the rough ER and the smooth ER. Now, in ER or the endoplasmic reticulum, okay, is the network of tubular and vesicular protein or vesicular structures which are interconnected with one another. Now, some parts are connected to the nuclear membrane while others are connected to the cell membrane. So two types, we have the smooth and the rough. Smooth in a sense that lacks ribosome, and rough because of studded with ribosomes. Now, function of ER gives internal support to the cytoplasm. Rough ER synthesize secretory proteins and membrane proteins. The smooth ER synthesize the lipids for cell membrane. Now, in liver cells, the smooth ER detoxify drugs and poisons. In muscle cells, the smooth ER store calcium ions. For the Golgi body. So the Golgi body, discovered by Camillo Golgi, formed by stack of 5 to 8 membranous sacs. Now, sacs are usually flattened and are called the cisternae. Has two ends. We have the cis phase situated near the ER or the endoplasmic reticulum, and the trans phase situated near the cell membrane. Functions: modifies, sorts, and packs materials synthesized in the cell. Delivers synthesized materials to various targets inside the cell and outside the cell. Produce vacuoles and secretory vesicles, then form plasma membrane and lysosome. So how Golgi bodies work? Now the Golgi bodies receive proteins and lipids from the rough ER. Okay, from the rough ER. Now it modifies some of them and sorts, then concentrates and packs into sealed droplets called the vesicles. Now this one, the vesicles, if they're ready for transport to their next destination. The next one is the lysosome. Small spherical single membrane sac found throughout the cell or the cytoplasm, filled with hydrolytic enzyme, occur in most animal cell and few in type of plant cells. Now, for the function of the lysosome, first, help in digesting of large molecules. Protect cell by destroying foreign invaders like bacteria and viruses. Degradation of the worn-out organelles, then in dead cells perform autolysis. The next one, we have the vacuoles. Now, vacuoles, we have single membrane sac filled with liquid or sop, okay, which contains the water, the sugar, and ions. In animal cells, vacuoles are temporarily small in size and few in numbers, while in plant cells, vacuoles are large and more in numbers. Okay, may be contractile or non-contractile. Now, functions: store various substances including waste products, maintain osmotic pressure of the cell, store food particles in amoeba cells, and provide turgidity and rigidity to plant cells. For the mitochondria, or the mitochondrion, the singular form of the mitochondria, is a small rod-shaped organelles bounded by two membranes. We have the inner and we have the outer membranes. Now, the outer membrane is smooth and encloses the contents of the mitochondria. The inner membrane is folded in the form of shelf-like inward projections called the cristae. Inner cavity is filled with matrix, which contains many enzymes. Contain their own DNA, which are responsible for many enzymatic actions. Now, the function of the mitochondria is to synthesize energy-rich compound known as ATP, where the ATP stands for adenosine triphosphate. Now, these ATP molecules provide energy for the vital activities of living cells. Plastids. Now, plastids are double membrane-bound organelles found inside plants in some algae. So they are responsible for activities related to making and storing food. They often contain different types of pigments that can change the color of the cell. Now, plastids can be chromoplast, chloroplast, or the leucoplast. So, for the chromoplast, these are plastids, heterogeneous organelles, responsible for the pigment synthesis and strange and specific photosynthetic eukaryotes. So the chloroplast is responsible for the photosynthesis, which contains the photosynthetic pigment called the chlorophyll, while the leucoplast are plastids that are considered to be colorless. So this includes the three types. Okay. So for the chromoplast are plastids that produce and store pigment. So they are responsible for different colors found in leaves, fruits, flowers, and vegetables. Example, carrot contain the pigment carotene. The mango contains the pigment xanthophyll, and the tomato contains the pigment lycopene. For the leucoplast are colorless plastids that store food. So they are found in storage organs such as fruits, tubers, and seeds. Example, we have here the potato tubers that contains the food starch. So the food is in a form of starch. The maize grains, we have the food is in the form of protein. And for the castor seeds, the food is in the form of oil. Now, the chloroplast is a double membrane-bound organelles found mainly in plant cells, Usually spherical or discoidal in shape, shows two distinct regions. We have the grana and the stroma. So, granum is the singular form and we have the stroma. So, the grana are stacks of the thylakoids. Okay, this one stack here is known as the thylakoid. So that set or stacks of the thylakoid is called the grana. Okay, thylakoids contain the chlorophyll molecules which are responsible for photosynthesis, and the stroma is the colorless dense fluid. So in the case of the photosynthesis, the light-dependent reaction happened in the thylakoid part of the chloroplast, while the light-independent or the dark reaction happened in the stroma part of the chloroplast. Now, the function is to convert light energy into chemical energy in the form of food. That is from radiant to chemical. Provide green color to leaves, stems, and vegetables. Next is we have the centrosome. So the centrosome is the membrane-bound organelle present near the nucleus. Consists of two structures called the centrioles, and centrioles are hollow cylindrical structures made of microtubules. Now, centrioles are arranged at right angles to each other. So function is to form spindle fibers which help in the movement of the chromosomes during cell division. So it also helps in the formation of the cilia and flagella. So the cytoskeleton formed by microtubules and microfilaments, and microtubules are hollow tubules made up of protein called the tubulin. So microfilaments are rod-shaped thin filaments made up of protein called actin. Now, functions determine the shape of the cell, give structural strength to the cell, and responsible for cellular movements. So the differences between the prokaryotic cell and the eukaryotic cell. Now, when we say prokaryotic cell, we're talking to a cell that is considered to be unicellular, while the eukaryotic one is multicellular cell or many-celled organism. Now, for the case of the prokaryotic, first we have there nucleus is underdeveloped, while in the eukaryotic, nucleus is well developed. Next is only one chromosome is present in the prokaryotic while more than one chromosomes are present in eukaryotic. Membrane-bound organelles are absent. Size ranges from 0.5 to 5 micrometers. And examples of prokaryotic cells, we have bacteria and blue-green algae. So while in the eukaryotic cell, membrane-bound organelles are present, size ranges from 5 to 100 micrometers, and examples are all other organisms except bacteria and the blue-green algae. So the animal cell comparison between the animal cell and the plant cell in terms of size, the animal cell is smaller than the plant cell. Cell wall is absent in the plant cell, in the animal cell, but cell wall is present in the plant cell. Now, plastids are absent in animal cell while plastids are present in the plant cell. So, vacuoles are smaller in size and less in number in animal, while plant vacuoles are larger in size because this one is for storage of food. Centrioles are present in animal while centrioles are absent in plant cell. Okay, I hope you learned something new today, and don't forget to take down notes and get ready for our next discussion. Okay, that's all.